Hey guys, so today we're going to be talking about viruses. So we're moving on to the next portion of these microorganisms. So some of the first things you need to know about viruses, as we have said before, is viruses are too small to be seen with even a light microscope and cannot be cultured outside of their hosts. Thus, viruses could not be studied until the 20th century, despite existing much longer than that and causing significant morbidity and mortality in other organisms, including humans. So the characteristics of viruses also begs the question of, are these viruses actually alive? So first of all, let's start with a definition of what life is. So life is an organismic state characterized by the capacity for four things. These are the metabolism, growth, reaction to stimuli, and reproduction. So if you look at these characteristics individually and look at, if they are present in viruses, if you look at metabolism, for example, so there's usually viruses do not have a metabolism. There are some exceptions, but the metabolism can never be independent of the host. Viruses also do not grow. They cannot react to stimuli. And yes, they can reproduce, but this is also limited to certain phases of their life cycle. So unless they are within the host cell, they cannot reproduce either. So this essentially means that the answer of whether they're alive or not is still debated hotly in the scientific community. So if you look at the continuum from non-living to living in the microbial world, so if you look at this figure here, you see a E. coli. So E. coli is typically about 2 microns by 1 micron. So it's very, very small. But even that is absolutely gigantic if you compare it to certain viruses. I mean, just look at how small the Mimi virus is. And if you look at the number of genes this Mimi virus has, for example, it has 911 genes, whereas the E. coli already has 4,478. But this is a rather large virus, where if you compare it to something like the HIV only has 9 genes. So it's a very, very, very short genetic or bit of genetic material that it has. So the question of whether viruses are living organisms essentially has a bit of an ambiguous answer. Because life can be defined as a complex set of processes resulting from the actions of proteins specified by nucleic acids. This is nothing new to you guys. And then the nucleic acids of living cells are in action essentially all the time. They're being utilized and giving instructions constantly. But because viruses are inert outside of a living host cell, in this sense, they are not considered living organisms because their nucleic acids are essentially non-functional unless they are in a host cell. But once they enter a host cell, the viral nucleic acids can become active and viral multiplication can actually occur. So in this sense, viruses are alive when they multiply in the host cell that they have infected. So there's no real conclusive answer. It very much depends on your point of view. So if you look from a clinical point of view also, viruses can be considered alive because they cause infection and disease, just as pathogenic bacteria, fungi, and protozoa and other microbes do. So depending on one's viewpoint, a virus can be regarded as an exceptionally complex aggregation of non-living chemicals. So you can see it as non-living, or it can just be seen as an exceptionally simple living organism. So there's no real definitive answer. It very much depends on how you view viruses. So how then do we define what a virus is? So viruses were originally distinguished from other infectious agents because they are, really, really small. They're filterable, and because they are obligatory intercellular parasites, meaning they absolutely require living host cells in order to multiply. However, both these properties are also shared by certain small bacteria, such as the rickettsias. And here again, you can see your number of bases and how viruses are very much on this small side of things. And that's also why they're parasitic, because they lack the genetic information to sustain their own livelihoods. And that's also why you find certain bacteria, for example, the rickettsias, on this side of the graph, where their number of bases is actually quite few, and thus they are parasitic. They can't survive without hosts. So you can see from all the organisms, whether you work in eukaryotes, bacteria, and archaea, if they have a small number of base pairs, they are typically parasitic, and then they move, as you increase their complexity, they can start becoming free living. But there is some overlap here between parasitic bacteria and some of your viruses. So here you can see a more clear breakdown of when we compare viruses and bacteria. So again, there's a continuation of similarities between viruses and smaller bacteria. Note, however, that there are still some differences because bacteria are, by their very natures, more complex than viruses. Something simple that you can look at is that the presence of ribosomes. So even in these small bacteria like the rickettsias and the chlamydias, you will still find ribosomes, where viruses lack them completely. So despite their similarities, there are still some differences. So how did we get to studying viruses? How did the scientific community evolve looking at, or change how they looked at viruses? So essentially, it started off with researchers not being able to imagine, submicroscopic particles. I mean, we did quite a comprehensive history of microorganisms, and just you guys are well aware at this point of how long it just took for us to establish that life was, there was life at the microscopic scale. So you can just imagine that researchers could just not fathom that something could exist smaller than microscopic scales. These were, of course, light microscopes back in the day. So initially, they described these infectious agents as contagium vivium fluidum, which is essentially Latin for contagious fluid. And this fluid nature of it came from that these viruses could not be removed by filtration. So it was thought that the infectious agent was actually contained within the fluid and wasn't a separate entity. And then by the 1930s, the scientific community started using the term virus, which is the Latin word for poison, actually, interestingly enough. So then we moved on to more, the discoverers of viruses. So the first person you should be aware of is, of course, Edward Jenner. He should ring some bells for you guys because he was the guy that also discovered vaccination using cowpox. Remember the vaccinia, where that word stems from, to give cross immunity to smallpox. So he infected the young boy with cowpox, which then made the boy immune to smallpox. And then moving on to the 1800s, we find Pasteur again, and he produced the first vaccine for rabies by growing the virus in rabbits and then weakening it by drying the affected nerve tissue. And then Ivanovsky was the first to actually purify a virus. This TMV you see over here is the tobacco mosaic virus. So this was, of course, a plant infecting virus. And then Stanley succeeded in the crystallization of this TMV. So there are some big names in the history of viruses that you should be aware of. Now we're going to be moving on to the general properties, where we will be looking at the sizes of these viruses, the life cycle, and then looking at stuff like virions and prions and the like. So first thing that you have to, again, just to stress this, that these organisms are, or if you want to call them organisms, viruses, maybe is a better, because we don't know if they're alive or not. So viruses are unfathomably small. They are ridiculously small. So viruses are tens to hundreds of nanometers, while bacteria are generally on the scale of thousands of nanometers or micrometers. So they are really, really small. So over here in the diagram, you can actually see a human red blood cell. And then you can compare that to E. coli, which is a small bacteria. and then just look at how small these viruses are by comparison, like the rhinovirus and bacteriophages, Veroids. They're really, really, really small. And so to give you some more perspective here, so humans are typically 100,000 times bigger than our cells, and 1,000,000 times bigger than a bacteria, and 10 million times bigger than an average virus. So if a virus was the size of a 5-cent coin, a bacterium would be the size of a dinner plate, and you would be 200 kilometers or 124 miles tall. So yeah, these things are really, really, really tiny. Which gives us to, stuff like this, where, the viruses reunion and like they were nano before it was cool to be nano. It's a little bacteriophage over here. Okay, so let's move on to some to a life cycle of these viruses. So there are some characteristics that you have to be aware of when it comes to viruses. So viruses typically contain a single type of nucleic acid. So they have either DNA or RNA. It's very rare that you find both in a virus. So they're typically classified as either DNA viruses or RNA viruses. They contain a protein coat. This is sometimes enclosed by an envelope of lipids, proteins, and carbohydrates that surrounds the nucleic acid. Viruses multiply inside living cells by using the synthesizing machinery of the cells. So you can think of it as the virus hijacking the cell's machinery. And they cause the synthesis of specialized structure that can transfer the viral nucleic acid to other cells. So they replicate themselves using the cell's replication machinery, and then that is how they can then be transferred to other cells. We'll look at that in a bit more depth here in a second. So you can essentially think of a virus life cycle of consisting of two separate phases based on whether they are inside the host cells or not. So if they are extracellular, meaning they haven't infected a host cells, they're typically classified as virions. So what is a virion? A virion is a complete, fully developed infectious viral particle composed of a nucleic acid that is surrounded by a protein coat on the outside of the host cell, and is a vehicle of transmission from one host cell to another. And then once these viruses actually infect a cell and enter the cell, they become obligate intercellular parasites. So that's the other phase that they can be in. And here you see a little breakdown of what a virion is. So it's a nucleic acid, it has its protein coat, and it also can have other structures such as an envelope, spikes, or tail. This varies by the type of virus you work with. So some general properties, again, of virions. So we'll see in the next couple of slides. I'm just going to give you a breakdown quickly of what you will be looking at. So first we'll be looking at helical virions. So they can either be enveloped, meaning they are contained within an envelope, well, a coating. There's another coating surrounding it. You'll see more examples of that later. So these virions can either be helical, so they'll make a little helix. They can be polyhedral, such as the polio virus, and you can also find enveloped polyhedral virus. This, like herpes, is an example of that. And they can also be complex, such as bacteriophages or the smallpox virus. So moving on, this is what a helical virus will look like. So you can see the little helix forming over there, and you have your nucleic acid in the middle, and then a capsid on the outside with the capsomeres forming your capsid. So an example of this would be the Ebola virus, which you can see there. It like twirls and whirls. Again, this is very, very small. So it's hard to find very high quality images of this. So helical viruses essentially resemble long rods that may be rigid or flexible, and the viral nucleic acid is found within a hollow cylindrical capsid that has a helical structure. That's what you can see over here. So the viruses that cause rabies and Ebola hemorrhagic fever are helical viruses. So now we're moving on to polyhedral viruses. So here you can see that the shape that it'll have, it looks completely different. So many animal and plant and bacterial viruses are polyhedral. So there are many-sided viruses. So the capsid of most polyhedral viruses is in the shape of an icosahedron. And that's an example of it over here, which is essentially a regular polyhedron with 20 triangular faces and 12 corners. So here you can see the 20 triangular faces. So I don't know, if any of you guys are fans of Dungeons and Dragons, or you play Magic the Gathering or something like that, but those D20 dices, that's an example of this. That gives you an idea of what you are looking at. Yeah. So the capsomeres of each face form an equilateral triangle. So these are all little equilateral triangles. So diseases that are associated with polyhedral viruses include many very common human infections that are estimated to be responsible for between 2 and 5% of all respiratory infections. They're usually mild respiratory, gastrointestinal, and eye infections, with the serotypes being of 3, 5, and 7. Feel free to read a bit more about that. It's very unlikely that I'll ask you something like that, as long as you know what these viruses look like and the differences between them and the other types, you should be okay. So the envelopes that I kept yammering on about earlier. So here you can see an example of an enveloped helical virus. So there you can see the helical virus inside, and it's essentially just contained within this envelope. And the same this side for an enveloped polyhedral virus. So there's the polyhedron, the polyhedral shape, and then it's also in an envelope that can have spikes on the outside. So essentially what happens here is that the capsid of some viruses is covered by an envelope. These envelope viruses are roughly spherical. So if you look at this virus without having the cross section that we have here to be able to see inside, it'll just look like a little sphere. I mean, this one's a bit more ovoid shape, but yeah. So so when helical or polyhedral viruses are enclosed by envelopes, they are called enveloped helical and enveloped polyhedral. And the simple examples here is influenza is an example of the enveloped helical virus, and herpes is an example of an enveloped polyhedral virus. And now we move on to complex viruses. I'm sure many of you have seen these bacteriophages before. So complex viruses essentially have additional structures beyond just a capsid and an envelope. You can see it includes tail fibers and a sheath and the like. So, and common examples of this would be bacteriophages. Bacteriophages are, of course, viruses that infect bacteria. So what I do want to point out in this figure, though, is notice that the head is polyhedral, and that the tail here is helical. And then when this virus, this bacteriophage, the DNA is actually contained within this polyhedral capsid or head of the virus. So continuing on with complex viruses, there's another example, and they are pox viruses. So they do not contain a clearly identifiable capsid, but have several coats around the nucleic acid, which you can see in this image over here. And then just as a note here, that oftentimes RNA viruses will have a reverse transcriptase enzyme. We'll be looking at that a bit more later on, but just keep in mind that the RNA viruses will typically have that enzyme. And that's, that's the enzyme that it will need to for replication. Okay. So continuing on here, you can also look at the genetic material, and that's also how you can characterize certain viruses. So there are viruses that have double-stranded DNA or dsDNA. HPV is an example of that. You can also have single-stranded DNA viruses or ssDNA viruses, so that's such as the human Parvo virus. So Parvo is an infection that is common and highly contagious in children, and it's sometimes called slap-cheeked disease because of the distinctive face rash that develops. Then you can also have double-stranded RNA viruses or dsRNA, such as your Colorado tick fever. And you can also have single-stranded RNA viruses or ssRNA viruses such as HIV. Remember, we looked earlier at how little genetic material is actually contained in an HIV virus. So that brings us to retroviruses. So remember when I mentioned that RNA viruses contain reverse transcriptases? So retroviruses is an example. They're an example of that. So they carry reverse transcriptase, which they can use. They use the viral RNA as a template to produce complementary double-stranded DNA. So the enzyme will also degrade the original viral RNA, and the name retrovirus is actually derived from the first letters of reverse transcriptase. And then the viral DNA is then integrated into the host cell chromosome as a provirus. So unlike prophages, though, provirus never comes out of the chromosome. And a good example of a provirus here is HIV again. So as a provirus, HIV is protected from the host immune system and antiviral drugs. You can just essentially think of it as being a little bit incognito, where it's unable to be removed because it's incorporated into the host cell's DNA. So as you can imagine, reverse transcriptase does exactly what it states. It moves from RNA to DNA, which is contrary to what transcriptase is, which goes from DNA to RNA. I hope you still recall those lectures. So that brings us to virus replication, or the Lytic cycle. We're going to be specifically looking at bacteriophages because they're a very good example. We will be talking about some animal viruses there then, because there is a few differences, one in particular that you should be aware of. So the Lytic cycle can be broken down into 5 steps, which is essentially absorption, which will be the attachment. I will be showing pictures of this on the next slide, so don't feel too lost at the moment. Then it's followed by penetration, where the virus will be uncoating its genetic material. Biosynthesis, where the nucleic acid and the capsid is actually produced of the following viruses. And then everything that has been produced will be assembled in the maturation phase, and then there will be a release. So this is characterized by burst time and a burst size. So burst time is essentially the time from attachment to when the cell lyses. That is your burst time. So for these 5 steps to happen, is your burst time. So the burst time of viruses typically, and bacteriophages, typically range between 20 to 40 minutes. Burst size, on the other hand, is just the number of viruses released from the cell, when at the burst time. So the burst size typically ranges from 50 to 200 viruses. So for every cell that lyses, you can see that there would be a massive increase in the number of viruses present in the host organism. If it's, for example, a higher organism like a person. If it's a bacterial community, same thing. Okay, so here we're going to spend some time on this slide because this is pretty important. So buckle up, guys. So first of all, you will find step number 1 over here. This is where attachment happens. So attachment is essentially after a chance collision between the phage particles and bacteria at attachment and adsorption. Remember, adsorption is to the surface, absorption is within. So adsorption occurs. During this process, an attachment site on the virus attaches to a complementary receptor site on the bacterial cell. This attachment is a chemical reaction in which weak bonds are formed between the attachment and the receptor sites. So bacteriophages, for example, will use the fibers at the end of their tails, which you can see over there, as attachment sites with complementary receptor sites located on the bacterial cell wall. Then following step 1 of attachment, we have penetration. So after the attachment, the bacteriophage will inject its DNA, so its nucleic acid, into the bacteria. You can see that happening over there. You can see the bacteriophage being or injecting its genetic material in there. By releasing an enzyme that's called the phage lysozyme. This breaks down a portion of the bacterial cell wall over there, or over here. Yeah, so it breaks down that portion of the cell wall, allowing for penetration to occur. And during penetration, the tail or sheath of the bacteria of the phage contracts, and then the tail core is driven through the cell wall. You can see how the tail core is kind of penetrating the cell wall over there. The tip of the core then reaches the plasma membrane. Again, you can see that over there. And the DNA from the bacteriophage's head passes through the tail core, through the plasma membrane, and enters the bacterial cell. So remember that when we looked earlier at these phages, they typically keep their genetic material in the head. Well, that is now pushed out throughout the little virus, throughout the phage, into the bacterial cell. The capsid will then remain on the outside of the cell. So it sticks around on the outside because it remains attached there, but it's hollow. It's not doing anything. And therefore, the phage particle functions like a hypodermic syringe. So you can really think of it as like a syringe, just like squirting out this genetic material into the cell, into the bacteria. So then we move on to biosynthesis. This is pretty interesting. So once the bacteriophage DNA has reached the cytoplasm of the host cell, the biosynthesis of viral nucleic acid and proteins occur. So essentially what happens, this is where the hijacking happens. So the host protein synthesis is stopped by virus-induced degradation of the host DNA. So the host DNA is kind of broken down and non-viable anymore. Then the viral proteins will interfere with transcription, or they can repress translation. You guys should all be aware of those processes at this point. So the phage then uses the host cell's nucleotides and several of its enzymes to synthesize many copies of the phage DNA. So after stopping the host cell's DNA replication, translation, transcription, the phage will actually start using the host cell's nucleotides that are currently not doing anything because that process has been stopped to copy itself multiple times. The host cell's ribosomes, enzymes, and amino acids are used for translation. So this is where it literally hijacks the little bacteria's cellular machinery, if you will, and just multiplies itself. So I do want to note something here. I'm not sure if this is in your textbooks or not, but note that the period during viral multiplication when complete infective virions are not yet present is called the eclipse period. So viral multiplication has been completed, but the infective virions are not yet present. It's called the eclipse period. May or may not appear in an exam or a class test. So then we have maturation. That's our next stage that we move on to following biosynthesis. This is over here. So during maturation, essentially the bacteriophage DNA and capsids are just assembled into complete virions. You can see here everything has been produced, and now it's just assembling everything to form these complete virions. And following this, we have release. Which is just this is the final stage of the multiplication, and this is where the host cell will lyse or burst. And release the virions. So the term lysis is generally used for this stage in the multiplication of phages, because in this case, the plasma membrane actually breaks open, it lyses. So the cell literally bursts. And then, these virions are spread throughout, if it's bacterial culture, into the rest of the culture or wherever. Okay. So just a quick overview here, just a breakdown of what we just discussed. First, we have attachment, and keep in mind, this is specifically the lytic cycle. It'll become important in a second here, lytic cycle. We have attachment, where the phage attaches by tail fibers to the host cell, followed by penetration, where the phage lysozyme opens the cell wall, the tail shield contracts to force the tail core and DNA into the cell. We have biosynthesis, this is the production of phage DNA and proteins. Maturation, which is just assembly of the phage particles that has been produced. Then you have release. This is where the lysozyme breaks open the cell wall. So this is where the thing just bursts, it pops. So this results in a one-step growth curve. So essentially here on your y-axis, you have number of virions, and here you have time in days. So essentially you will start off with some virions, but then they will disappear in this eclipse period. So the virions at this point have attached to host cells and are busy with the lytic cycle. So you can't see them because they are inside the host cells. And then following the eclipse period, you'll have the virions being released and the cells lysing. And when that reaches a maximum, you will have acute infection. This is where the largest number of virions will be present. And this graph was specifically obtained from looking at data from cell cultures. So essentially, no new infective virions are found in a culture until after biosynthesis and maturation have taken place. So remember, their initial virion will inject its genetic material, and then there will be a lack of virions. You won't be able to find any virions even inside of cells because they haven't started assembling yet. And then most of the cells that are injected will die as a result of infection, and consequently new virions will not be produced. That's what happens over here. So at acute infection, after you've used up all the host cells that are present in a cell culture, you can't have any more virions being produced because they are obligate parasites. They need other cells. In this case, if you're looking at phages, they need other bacterial cells, otherwise they can't increase their numbers. So now we're going to move on to lysogeny. So in contrast to T-even bacteriophages, which are the bacteriophages we've been looking at, some viruses do not cause cells to lyse, and they also do not cause the death of the host cell when they multiply. So this is where we get to lysogenic phages, or the lysogenic cycles. So just as a note though, these lysogenic phages that are also called temperate phages can proceed through a lytic cycle, but they're also capable of incorporating their DNA into a host cell's DNA to begin the lysogenic cycle. So a completely different cycle, but they are linked together, where many viruses can use one for a while and then go on to the other cycle, and vice versa. So just the take-home message from the lysogeny. So in lysogeny, the phage remains latent. It's completely inactive, with the participating bacterial host cells known as lysogenic cells. So there are some results that you should be aware of from this type of replication. So first of all, you will have an immunity to infection by other phages. The host can exhibit new properties. This can include the formation of cancerous cells, and then you will see specialized transduction. So here you have a breakdown of both cycles. We just discussed the lytic cycle. Remember, lytic or this is where the cell will lyse and will split open at the end, but it can also, viruses can also form a lysogenic cycle. So here, if you go from number one, you will still have the phage attaching to the host cell, injecting its its viral DNA or RNA into the host organism. But here you find the difference, because you can now you can either start assembling new Veroids, or what can happen is in the lysogenic cycle, the circle DNA can recombine with and become part of the circular bacterial DNA. So see, there's the prophage that has now become part of the DNA of the host cell. And this is specifically, so the inserted phage DNA is called a prophage once it is incorporated into the host cell's DNA. So most of the prophage genes are then repressed by two repressor proteins, stopping the transcription of all other phage genes by binding to the operators. So thus the phage genes that would otherwise direct the synthesis and the release of new virions are essentially turned off. It's not active. And this is similar to, if you guys can recall, how the genes of the E. coli lac operon are turned off by the lac repressor. It's a similar process, not exactly the same, but it's similar. And then every time now, what would happen is every time the host cells goes through its its DNA replication. So every time the host cell's machinery replicates the bacterial chromosome, it will also replicate the prophage DNA. And this prophage will remain latent. So essentially what you can think of this now is every daughter cell of this bacteria will now have this prophage DNA still contained within its nucleic acid, so within its DNA. So it'll replicate and replicate and replicate and replicate, and completely unintentionally be also replicating the viral DNA that it's containing, the prophage DNA. And then some rare or spontaneous event, such as the action of UV light or certain chemicals, can lead to excision. So this is essentially the process of the prophage popping out. You can kind of think of it as that you have this circle that contains the prophage DNA, and that's now popping out. It's moving out. Once this happens, the virus can revert to the lytic cycle. So you will now be in this exact same position. Remember, though, this will not be the same organism. This will be some daughter, most likely will be some daughter cell of the original host cell that you started off with, because there is replication happening there. So you can have your original cell, and up until this point, it'll go through replication. So this is some bacterial daughter cell. It is not the exact cell that you started off with, although that one will still, the daughter cells, all the daughter cells can go through this process, and all the daughter cells, or the granddaughter cells, can also go through this process because it has replicated. All of them contain that prophage DNA because of the original infection. But anyway, so once this excision occurs, the a virus can revert to the lytic cycle, which will then lead to the release of new Veroids and the lysing of the cell. Okay. So now we're going to be moving on to the cultivation of viruses. So if you want to cultivate bacteriophages, you can use plaques, and then you can enumerate them from these plaques. I will show you a brief example and explain very, very briefly about that process. And then you can also, in animal viruses, remember these viruses need to be within their host cells. So if it's an animal virus, you're going to need to incubate and cultivate it, cultivate it inside living animals or embryonated eggs or cells or tissues, tissue cultures. So this is an example of what the plaques look like. Remember, this is for bacteriophages specifically. So essentially, these bacteriophages can be grown either in suspensions of bacteria, in liquid media, or in bacterial cultures on solid media. Give me a second here. So essentially, a sample of a bacteriophage is then mixed in with host bacteria and melted agar, and then poured into a petri plate containing a hardened layer of agar growth medium. Each virus will then infect the bacterium, will multiply, and then release several hundred new viruses, which will infect other bacteria, and in the immediate vicinity, producing more viruses, and so on and so on and so on. And following several multiplication cycles, the bacteria in the area surrounding the original viruses will be destroyed. Remember, they lyse at the end of the process. So this will produce a number of clearings that you can see over here, these little clear spots. All these spots are little clearings. This is where the bacterial cells have been destroyed. And you can definitely see them quite clearly on this lawn of agar, on this lawn of bacterial growth on the agar is probably more correct. And then, so each plaque here theoretically corresponds to a single virus in the initial concentration. There are some assumptions present there, but that's, again, these things are really, really small. So therefore, the concentrations of viral suspensions measured by this number of plaque, measured by this plaque technique, are usually given in the forms of, in terms of plaque forming units, so PFUs. So then moving on to some animal viruses, again, you need to grow these within living animals, embryonated eggs or cell cultures or the like. And there are some, viruses that you need actually a living organism for, of course. So that's typically where we use mice or rabbits or guinea pigs. Most experiments to study the immune system's response to viral infections must also be performed in virally infected live animals. That's the only way you can do it. Animal inoculation may be used as a diagnostic procedure for identifying and isolating a virus from a clinical specimen. And after the animal is inoculated with the specimen, the animal is observed for signs of disease, or the animal might be killed so that the infected tissues can be examined for the virus. Of course, we also, viruses are not limited to animals. So animals and plant viruses can also be grown in cell culture. So where continuous cell lines may be maintained indefinitely, again, as long as you keep adding cells, you could keep repeating this process. So just this is a simplification of the process, but essentially you have tissue that is treated with enzymes to separate the cells. The cells are suspended in a culture medium, and then normal cells or primary cells grow in a monolayer across the glass plastic container, and transformed cells or continuous cell cultures do not grow in a monolayer. So they essentially make these little clumps. In this context, it's important to know that transformed cells refers to the changes in cells that occur after incorporation of viral DNA. And then oftentimes lysogenic viruses can change cell properties, causing them to grow differently. So they can form tumors, for example. Okay, so animal virus life cycles. So now we're not discussing phages anymore. We're going to be moving on to animal viruses. And their lytic cycle is very, very similar to a bacteriophage, but there are some differences. We're also going to be discussing latent infections, persistent infections, and transformation. So starting off with what the difference is, most of this is the same, so I'm not going to, or is at least very, very similar, so I'm not going to go through too much detail for you. Feel free to read in your textbooks. Always a good idea to do that. But the major difference that you'll notice is that there is an additional step of uncoating. And uncoating here is the separation of the viral nucleic acid from its protein coat once the virion is enclosed within the vesicle. So the capsid is digested when the cell attempts to digest the vesicle's contents, or the non-enveloped capsid may be released into the cytoplasm of the host cell. So that's the primary, or maybe the most obvious difference. So you can this process, if you look at the attachment, penetration, and uncoating, there's one process you can look at called endocytosis, or at least receptor-mediated endocytosis. So this is just I think it's better to have you guys visually look at this. So this is the animal virus initially, and now it's attached to the cell, the animal cell that it's infecting. It produces the vesicle, which then follows it into the cell or enters into the cell. But once this vesicle is dissolved, you have a complete dissolution of everything except the genetic material. So just a little bit of a throwback here, challenge for you guys. What image do you guys think this is? Where did we get these images from? I'm just going to give you like 2 seconds. So this is actually a TEM image. I hope you guys can still remember back to the microscopy. Again, remember the very, very small sizes we're working with. Now, there's another approach that you can look at, which is fusion. So here, spikes attach to the host cell receptors and mediate endocytosis or fusion. Here you can see the spikes that is binding to the receptors, and then the virus will fuse to the host cell. So here you can see this little new area developing, or a bulge developing, where the other one was like an indentation that was formed. And then once this has been incorporated into the cells, into the host cell, again, you will find that the genetic material, the virion contents, has entered the cytoplasm. And again, once inside, the virus will hijack the cell's machinery, produce viral DNA and proteins that assemble new viruses. And biosynthesis and maturation are very, very similar to bacteriophages. So I'm not going to be discussing them. So what happens? How do these viruses then how are they released? How are enveloped viruses released? Because you can, of course, imagine that if this thing just bursts open, you will struggle to form an envelope. So after the sequence of attachment, entry, uncoating, and biosynthesis of viral nucleic acids and proteins, the assembled capsid containing the nucleic acid pushes through the plasma membrane. So you can see it pushing through, and it actually takes a portion of the plasma membrane, and that is what the envelope is formed from. And once this plasma membrane forms part of the virus external structure, that is when it is known as an envelope. So notice specifically how the envelope virus uses the plasma membrane during release. So it makes this little bud. You can see one over there too, from the alpha virus. It makes this little bud, and at some point it'll just plop off, and then the plasma membrane will be its envelope. And this extrusion of the virus from the host cell is one method of release that doesn't result in the cell bursting. And budding of, like I just said, does not immediately kill this host cell, and in some cases the host cell can survive. Now we're going to be discussing latent infections. So what is a latent infection? So a latent infection is a type of persistent viral infection which is distinguished from chronic virus infection. Latency is a phase in certain viruses' life cycles in which, after initial infection, proliferation of virus and proliferation, there's a cessation of proliferation. So just to simplify that, so after initial infection, the proliferation or the creation of more virus particles will stop completely. But the viral genome is not completely eradicated. And some of these latent viruses can actually integrate their DNA into the host genome, while others they don't do that, but some can. So it's very similar to the lysogenic cycle of phages. Remember how the phages in the lysogenic cycle also incorporated their DNA into the host DNA, but it's questionable whether viral nucleic acid integrates into the host genome. That is not, it can or it cannot happen. So examples here can include cold sores, chickenpox, or infectious mononucleosis. Here we're going to look at shingles, which is also an example of a latent infection, which is caused by the Varicella-Zoster virus, which is the same virus that causes chickenpox. And approximately 10 to 20% of people who've had chickenpox will get shingles in some point of their life. We're moving on to persistent infections. I might be just speeding up a little because we are we still have quite a few slides to go. So persistent infections are, different from latent viral infections in that the detectable virus builds slowly over a long period rather than just appearing suddenly. This becomes very apparent on the following slide. So a good example here is the Hepatitis B virus, where symptoms are variable and include yellowing of the eyes, abdominal pain, and dark urine. Your kidneys essentially failing. Some people, particularly children, don't experience any symptoms. However, in chronic cases, can result in liver failure, cancer, or scarring. So it's your liver failing, sorry, not your kidneys. I think I said kidneys. Another example is the Epstein-Barr virus, which is responsible for mononucleosis. And after the initial bout of fever, sore throat, and swollen lymph glands, the virus establishes a dormant infection in which the viral genome persists in the cells of the immune system. And this periodically, you can see this infection being reactivated and sort of flaring up. And infection variants are shed in the absence of clinical symptoms. These reactivations lead to the transmission of infection to new hosts. Okay. So this is, if you look at the number of virions and how these infections differ. So during an acute infection, there's your eclipse period, and then you'll just have this massive spike, and that's your acute infection. That's usually just, a couple of days after your initial infection or initial exposure to the viroid or the virus. However, over a persistent infection, remember they stated that you can have this slow buildup, which then eventually leads to a large increase, where a latent infection is just not present at all until it just suddenly explodes or is suddenly just there. So yeah, during a persistent infection, the viral load will increase gradually over time. You can see it over there. And with a latent infection, the outbreak occurs completely suddenly. So there's no slow onset to them. And some persistent viral infections are fatal, such as HIV and some cancer-causing viruses. Sorry. Okay, and then continuing on with our animal virus life cycles, we'll be looking at transformation, which is essentially the conversion of normal host cells into tumor cells, also called neoplasms. So a neoplasm is a new or abnormal growth of tissue in some part of the body, especially as a characteristic of cancer. And then it's essentially contact inhibition is a growth mechanism which functions to keep cells growing in one layer thick cell, a monolayer, and then when that is compromised, you find these clumps of cells. Remember, we spoke about that earlier too. And it's interesting to note here that approximately 10% of cancers are viral induced. These include cervical, anal, and liver cancers. So oncogenic viruses, oncogenic implies cancer causing. You can see there's Adenoviridae, which is a mild respiratory infection in young children known as, the common cold, that can lead to life-threatening multi-organ disease in people with a weakened immune system. You get your Papoviridae, which includes HPV. You get Hepatitis which includes hepatitis B, and on the RNA side of things with your retroviruses, remember there's that little diagram we discussed earlier where the cells are not growing in a monolayer anymore, and that is indicative of the cancer forming, where it's just this clump of cells. On the RNA side, human T-cell lymphotrophic virus, or Htlv, is a family of viruses that are caused by human retroviruses, and they can cause a type of cancer called adult T-cell leukemia or lymphoma. We're also going to be now looking at some unconventional viruses, and these can lead to slow viral infection, which is essentially a disease process that occurs gradually over a long period of time, and it's usually fatal. Examples here include the measles virus, which can, after several years of the initial infection, you can cause severe encephalitis. And here you can see the number of cases of measles per year in the United States, where it was essentially, a non-issue until a certain movement started, and then now you have a massive spike in measles cases. And this movement I'm referring to is the anti-vax movement, of course. I'll have a bit to say about them at the end of the lecture. Now moving on to prions, and this is actually pronounced prion because there are, it's quite a confusing term because there are two other things that are called prions, and there's also called a prion, but this is specifically pronounced prions, and it stands for proteinaceous infectious particles. So they contain a protein and reproduce inside cells, but they seem to have no nucleic acid. So examples here include mad cow disease as an example. Moving on to Veroids. So these are short pieces of naked RNA. They have no protein coat. They typically can cause plant diseases, such as host plants, such as potatoes and tomatoes. And this is what a structure of a Veroid looks like. It's a circular single-stranded RNA with some pairing between complementary bases and loops where no such pairing occurs. So here we get to what I was discussing earlier. There's quite a lot of noise in the media and quite a lot of attention about the anti-vax movement and anti-vaccination. And I think something that I would like to just talk to you about here for a bit is what started these rumors. So essentially, Lancet published a paper by Dr. Andrew Wakefield. Notably, he has had his degree revoked since, which was a dramatic study that found a connection between autism and vaccines. But very, very importantly, because a lot of anti-vaxxers like to refer to this thing, but the problem is This study had some major flaws. It was not based on any real statistics. It did not have a control group. It relied on people's memories, which is a notably and a known source of misinformation, because people's memories aren't that great. And it made vague conclusions that were not statistically valid. So the problem was also, of course, these rumors were then taken and run with by certain celebrities and people who have no idea what they're talking about, or who have just been caught up in this wave, this hype wave. And social media actually, obviously furthers this problem. So yeah, celebrity noise definitely took this idea. Celebrities took this idea and ran with it, and that's why it gained so much traction. And that's unfortunately leading to the resurgence of some diseases that we had essentially all but eradicated globally. So the take home is that vaccines do not cause autism. The debate is completely over. It was rejected by the medical community, by the scientific community, by the research community, by people with autism, and even the autistic community. And this myth is just completely busted. It's similar to how, the climate change thing. It's very much settled science. Calling it a debate at this point is a big misnomer, because it is not a debate anymore. It is a given. It's an accepted fact in the scientific community that vaccines do not cause autism. But not vaccinating your kids can definitely lead to the resurgence of some diseases, and it will impact herd immunity, where people who are not able to take these vaccines or get these vaccines will be susceptible to stuff like measles, like we discussed earlier. Anyway, my little tangent aside, let's look at some review questions. So, true or false, viroids are short pieces of naked DNA and have no protein coat? I'll give you guys about 5 seconds for this one. The answer here is, of course, this is false, because viroids are pieces of RNA, not DNA. True or false, the Lytic cycle comprises 4 steps. Was it 4 steps in the Lytic cycle? The answer, of course, here is false. Remember, there are 5 steps. It's adsorption or attachment, penetration, biosynthesis, maturation, and release. 5 steps. Moving on to review question number 3. Viruses can easily be removed from liquids through the process of filtration. The answer here is false. Remember how ridiculously small these viruses are, and that's why they were originally called, infectious fluid or poisonous fluid, because it was deemed, you simply can't remove them due to their small size, not that way at least. And then, true or false, viruses are able to respond to stimuli. Can they respond to stimuli? And the answer here is false. Remember again how simplistic these things are and how they lack the genetic material to be able to respond to stimuli. Okay, guys, that'll do it for today. And I will see you guys in the next lecture. Cheers.